Okay, so I want to go through mitosis and meiosis one more time. And this time, instead of drawing all the stages, I'm going to show you the most important ones that will you can focus on to make sure you understand these two processes. So let's start with a cell. And we're going to do our typical n equals 4. Which means you're going to have 4 chromosomes. Oh, that's not a very good color. 4 chromosomes in your cell. Okay. So this cell is at G1 phase. For both mitosis and meiosis, the cell will go through S phase. So you remember that means DNA replication happens. And even though technically we cannot see the chromosomes until prophase, we always just draw them as if we could see them, because otherwise it would be like this. Just a bunch of squiggles in it, and it doesn't really show us much. Okay, so we've gone through S phase. We're in G2, and now we're ready to do mitosis or meiosis. So on this side of the page, I'm going to show you mitosis. Right? So if a cell's going to go into mitosis, right, you know that in prophase, you can start to see the chromosomes and the nuclei, the nuclear membrane envelope, let's call it properly, disappears. Okay, so I'm not going to bother drawing it because it looks pretty much like what we have here. The most important phase to get right is metaphase. Okay, so actually, let me move my arrow down here. So, or I guess it's going through prophase. That's where it's starting. What we're going to first draw is metaphase. And what is important in mitosis metaphase is that you line up all the chromosomes right down the middle. Okay. Line up, down, middle, to split the sisters. Okay. So if we drew, that's pretty light. If we drew our spindle fibers, remember that the spindle fibers are connecting to, I don't know why I'm trying to draw with that, to the centromere region via the kinetochore proteins, and outside here are the centrosomes, where the centrioles have started generating those spindle fibers, which are microtubules, part of the cytoskeleton. So I'll just write microtubules, spindle fibers, and they do the pulling and pushing to get the chromosomes to their proper location. So anaphase is the splitting of them, telophase is generating the new nuclei, cytokinesis is the splitting of the cytoplasm. Sorry, I have a hard time drawing and talking. And what you're left with is 2 identical cells, right? So if you can split, if we look at this in metaphase, if you can split that correctly, I'm going to try to use this. If you line up the chromosomes correctly, all you're doing is splitting down the center. No, that's really faint. I hate to use this. Right? All you're doing is splitting down the center so that your 2 cells now have one of each colored chromosome. They're identical to each other, and they're identical to the cell that we started with. Mitosis. Meiosis It's just a little bit different. Okay. So meiosis also has prophase, where you see, so I'm just going to put here ana and telo and cytokinesis all happened. Okay. We're not going to draw them all out because it just takes a lot of time, as long as you're understanding how the separation of chromatids happens. So meiosis, you have prophase, same as before. This time when we go to metaphase, we line up the pairs. So I'm going to pair, in my case, green and orange, and red and blue. And it doesn't matter which side you put who on. Okay. Now, technically, yeah, I should be more precise. Okay. So, crossing over is happening in prophase 1. Okay. And then, so you could draw them anywhere in the cell. I just by habit get them already lined up. Okay. So let's do this. So crossing over is how we lined them up, or how we put them together. Metaphase, let's do the whole exchange thing. So we're exchanging the tips. Right? And the homologous pairs are lined up down the middle, which is different than metaphase. So I want you just to compare these two. Okay, so metaphase, they first line up right down the middle. In metaphase 1 of meiosis, the homologous pairs line up, and what you're going to do when you draw those spindle fibers is you're going to split right down the middle. Okay, so you haven't separated any sisters yet. You're separating the homologous pairs. Okay, so you could draw out anaphase where they're moving, and telophase where they're starting to reform the nucleus, and cytokinesis, but I'm just going to jump straight to the end result, and I'm going to line them up so that they are ready for metaphase 2. So once you get them lined up correctly in metaphase 1, all you're doing is splitting them down the middle, and then you're going to split them down the middle again. Okay, so metaphase 2, right? Now we're going to split the sisters. And so we're splitting these chromosomes right down the middle to make 4 genetically, sorry, my chromosomes are coming apart a little bit here. Let's see if I can get orange, and this has some orange, and this has orange tips. That one has green tips, and that one's a full green. All right, so let's see. I have 2 chromosomes, so N equals 2. I have 1 from each homologous pair in every gamete, and they're all a little bit different, so they're all genetically unique. Okay. So I know it looks simplified, and that's really all you need to remember to split the chromosomes correctly. Metaphase, I'm going to get rid of this black line for a second. Whoops. I'll stop. Sorry, I lost a chromosome. All right, metaphase, you line them up individually down the middle so you can split the sisters. Meiosis 1, you line them up so you can split the homologous pairs. I knew that was going to happen. And meiosis 2, you split those sisters. Okay, so practice this. You've got some practice homework that you can do if you choose, and it will have you drawing these out. Okay, let's talk about the differences between mitosis and meiosis. A big difference is should use a different color. Mitosis, you're producing 2, so you're going from 1, let's do this, 1 diploid to 2 diploid cells. Meiosis 1, or meiosis, you're going from 1 diploid to 4 haploid cells, right? Mitosis, you're making genetically identical cells. Meiosis, you're making 4 genetically unique cells. Okay. This process of going from diploid to haploid is called genetic reduction or chromosome reduction because you're cutting the total number of chromosomes in half at the end of meiosis. Another difference is this has 1 round of cell division. Meiosis has 2 rounds of cell division. Okay. So I want you to think about some of the similarities for your post-quiz short answer. Okay. There are some videos you can watch. This is links to the video above that compares meiosis and mitosis. We didn't get a chance to do these, but these were your clicker questions. So I'd suggest you pause the video for a second, answer the clicker questions, see if you get them right. So when do homologous chromosomes move toward the opposite pole? And that's meaning these guys. And so that is meiosis 1. Oops, I'm not sure I'm on the right thing. Okay, if the DNA content of a cell in G1 is Z, just making up a number, what is going to be in metaphase of meiosis 1? And the answer is 2Z, right? Because you've duplicated that chromosome. This is really not fun without you guys. Meiosis 2 typically produces blank cells, each of which are probably not good English there. So meiosis 2 will produce 4 cells, right, that are genetically non-identical. Synapsis. This is another word for crossing over. When does that happen? It only happens in prophase of meiosis 1, or I should say primarily. And here's one that's a really good one to discuss in class, so I'm bummed that we didn't get to do this. Which of the following statements is not true? Okay, so you should know diploid cells can divide by mitosis. That's what we've been doing. Diploid cells can divide by meiosis. That's what we've been doing. Haploid, oh, this is not a, This is not a good question. Oh, maybe anyways, okay, we'll just walk through this. Haploid cells can divide by mitosis. This is true. So with bacteria, although they don't divide by mitosis, so I should say with, like an amoeba, and I always spell amoeba wrong. I know there's an, I don't know. Okay. So if it had a chromosome, it would replicate that chromosome and then divide into 2 amoebas. Okay? Even if it's haploid, meaning it has only 1 copy of the chromosome. Haploid cells cannot divide by meiosis. Somehow I reworded this and messed it up. Because I added this. That's different than yours. Haploid cells cannot divide by meiosis because you cannot go from 1 chromosome to cells with half a chromosome. That would not work, right? So you've got to be diploid to be able to divide by meiosis. Okay. Another concept we kind of talked about, but we're going to expand on, is changes in chromosome number. So we're going to call this the normal diploid, 2N. Some organisms can actually have multiple copies of every chromosome. So your ploidy number is 2N. This is showing a fruit fly that is 3N. And I wrote 2N. Is 3N, which means it has 3 sets of every chromosome. Something that's tetraploid has 4 sets of every chromosome. What we talked about one of the earlier days is the term aneuploidy. And aneuploidy means changes in 1 chromosome, not the whole set. Okay? So trisomy means 1 chromosome has 3 copies. And monosomy means 1 chromosome has 1 copy. Okay? Compared to the normal diploid. So how does this happen? How does trisomy and monosomy happen? It's something called non-disjunction. Okay? And non-disjunction basically means incorrect separation of chromosomes. And we excuse me, primarily focus on non-disjunction in meiosis because it's going to affect the next generation. So if you get some non-disjunction happening in mitosis, that's only going to affect the cells that it generates. So maybe you have some funky skin cells. Most of the time, the cells will just die. But we can pass on changes in chromosome number through our gametes. Right? So remember that meiosis produces gametes. And so what can happen is you have incorrect separation either in meiosis 1. Okay? So instead of correctly separating the pairs, one of the pairs gets stuck together, which means this chromosome is going to have extra in from meiosis 2, and I'm sorry, this cell is going to have extra chromosome of the long one, and this cell is going to be missing chromosomes. So the final number, instead of just being normal haploid, this one has haploid plus 1, and this has haploid plus 2. So You're not going to have to draw this out. You will in genetics, but I wanted you to understand the concept of non-disjunction that causes aneuploidy. You can also have non-disjunction in meiosis too. So we've split the homologous pairs correctly, but then when we go to split the sisters, something happens, which means half of our gametes turn out normal number, and half of our gametes have either an extra or one less chromosome. So if it helps, draw this out, but it's either happening in meiosis 1 where you don't separate a pair, or meiosis 2 where you don't separate a set of sisters. And this is just showing you the aneuploidy conditions that can give result in live birth in humans. So trisomy 1, Down syndrome, you have 3 of chromosome 21. Trisomy 18, you have 3 of 18. Trisomy 18 and trisomy 13, the children usually don't live past a couple years of age, but and I'm going to forget his name. One of the politicians that was running for president a while ago, darn it, his daughter had Edwards syndrome, or has Edwards, and I think she's like 8 or 9 now, so she's lived a long time with 3 copies of chromosome 18. In the sex chromosomes, you can have XXY males, so your non-disjunction would be happening in the sex chromosomes versus these are the autosomes. You can have XYY males. You can have XXX, triple X females. The only monosomy that produces a live birth is what we call XO. So that's one X chromosome. We put the O there to make sure you haven't forgot something. So we're saying yes, they only have an X, they have no other sex chromosome. Aneuploidy occurs whether it's autosome or sex chromosomes the same way through non-disjunction. So where does this all come into play, right? So your meiosis is happening in your germline cells. Oops. Oh. Okay, that pen doesn't want to work. Germline cells, these are diploid, right? They produce your gametes, egg or sperm, that are haploid, and then an egg and sperm come together through what we call fertilization and make a new diploid zygote. So if each gamete has 23 plus 23, oh, something's weird happening, gives us back our 46, and that's called a zygote. And then you basically undergo mitosis, 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 mitosis. You become a little human being, and then eventually you produce ovary and testes cells. These are your germline cells, and those cells will undergo meiosis to produce egg and sperm. Okay. So mitosis is how you grow and repair and become a big multicellular organism. Well, I would love to ask you if you have questions, but we're not together. So let's keep moving on. For a cell 2n equals 4, which figure shows metaphase of mitosis? I really want you to think about this. I really want you to pause and make sure you can understand what these different images are showing you. So the correct answer is D, because you have all 4 chromosomes lined up in the middle for metaphase. Okay, so now this says for a cell with 2n equals 4, which shows meiosis 2. And meiosis 2 is going to be shown by B, where you are now splitting the sisters, and you can tell that it's meiosis because they have, if 2n equals 4, they've already done one separation. So even if you didn't get to see this cell, you would see, okay, I've got half the number of chromosomes. Normally, this must be the second phase of meiosis. And you can tell it's metaphase because the chromosomes are lined up and you're going to split the sisters. So, which one would be meiosis 1 metaphase? And that would be A. So if you can see there, you've really got XX, XX, right? So the homologous pairs are going to separate. So, what is this? So this is telophase 2, and all we need to do is cytokinesis and split that cytoplasm. All right, one more little, oops, that's not that. I wanted to talk to you for 1 second, well, not 1 second, but a couple minutes about the X and Y chromosomes. So we're talking humans. Females are XX, males are XY, and yes, the Y chromosome is a lot smaller. It's about 1/3 of the size of an X chromosome. You can see lots of genes on the X, some important genes on the Y. If we zoom in here, and I'll just warn you, this isn't going to be funny because we're not together, but I found this really funny one of the genes on the Y chromosome, like channel flipping, you know, air guitar. Okay, so this is really sexist. Sitting on the john, toilet, selective hearing loss. All right, so it was really funny, going to be funny in person, not so funny in a lecture slide, but I already had it there, so. What's really on the Y chromosome, and the most important one, is this, sex-determining region Y, which is called SRY. That makes you a male. That part makes you have male genitalia. Okay, you can see that the Y chromosome is 50 million base pairs. The X chromosome is 153 million base pairs. Lots of genes, and remember, every human has at least one X chromosome. Okay? Every human has at least one X chromosome. So you cannot live without the X. You can live without the Y. I already showed you a slide that had the aneuploidies for sex chromosomes, so an extra X or an extra Y. And then we kind of talked about this, like how could you be an XX male or an XY female? And like I said, the most important is that SRY gene. So if you have SRY, and even if it gets somehow put on the X chromosome, you are male. Okay? If you are missing the SRY, okay, even if you have a Y chromosome, but that part is mutated or missing, you are a female. So that's how sometimes chromosomes don't tell us the whole story, just looking at a karyotype, because a karyotype, we cannot see mutations or gene changes. The last slide I wanted to show you was just how interesting I think it is with chromosome number. So this is haploid number. So there's dogs. Dogs have 39 pairs. So I was off a little bit. So what is that? 78 chromosomes. Okay. And their genome size is basically 2.5 billion base pairs. Humans have 23 chromosomes, so 23 pairs, sorry, 46 chromosomes. And look we actually have a larger genome size. So the number of chromosomes does not necessarily tell you how many DNA nucleotides there are. Look at possums. Possums have 11 pairs and an even bigger genome size than humans. But genome size doesn't really matter because what might be more important is the number of genes. And unfortunately, I couldn't get everything to line up here, but we do have dog, right? So they say about 19,000 genes, right? A little bit smaller genome. Humans, about 22,000 genes. Even though our genome is larger, we don't have that many more. And then you can look at Arabidopsis, which is not on here. So Arabidopsis is 2n equals 10, so it has 5 pairs, really small genome. Potentially more genes than humans have, with a much smaller, like 10-fold smaller genome size than, sorry, my answering machine's going off. Smaller genome size than humans. So you cannot equate genome size to complexity, to number of genes. It all depends on what else is in that genome. And like I mentioned in class, in humans, only about 3 to 5% of these 3 billion base pairs encode for proteins. So we're trying to figure out what the other 95% does. All right, so there's chromosomes, mitosis, meiosis. Practice drawing them, getting them done, and enjoy your Thanksgiving.